thank you very much for this nice introduction. And I think the purpose with this presentation is to put your technologies produced by Fluicell in a context, how we use them actually in our, you can say, research based on what we do here in our pharmacological department. So in this case, we will talk about antibody treatment of pain or novel antibodies produced by Oblique Therapeutics, also in Gothenburg. And I will just start to give a short presentation of my group here. So as Edward said, I am head of the section of Translational Pharmacology, and we are in Biomedicum here. You see this fancy building here, which is constructed based on Marriott Hotel, actually, with 10 levels. So we have moved in here quite recently, 2018. And the purpose, we have 11 group members, mostly senior group members, researchers, but also a PhD student, Oskar Ljungholm, and another student. So the purpose with our research is in general to study mechanism of actions underlying pharmacological treatment of different diseases. For example, pain and cancer. I've also been working a lot with neuropsychiatric diseases like schizophrenia and depression, and have investigated antipsychotic and antidepressant drugs. We have several collaborations, and as I mentioned, we collaborate with Oblique Therapeutics. They are producing, I will talk about their platform for producing these antibodies that we are studying. We also are collaborating with the Royal Institute of Technology here in Stockholm and SLU in Uppsala, and of course Fluicell. So I have local collaborations, for example, with Professor Camilla Svensson. She's in charge with the laboratory. They are focusing on pain as well for treatment of rheumatoid arthritis. So we are collaborating with several associates here. So before we start to talk about the specific project, then I will just give a background concerning antibody therapeutics. So this field is growing rapidly, much due to the clinical success seen in with several monoclonal antibody therapies. So for example, you know about Herceptin, targeting the human epidermal growth factor receptor 2 on breast cancer cells. That's been very successful. We also have Humira, which is a TNF receptor antagonist, and it's mainly used for arthritis diseases. And then other examples are Avastin for cancers and eye disease, and alirocumab which is a cholesterol-lowering monoclonal antibody treatment strategy. Also, which is very interesting, is that due to advances in protein engineering, a wide variety of engineered antibody fragments have been also derived. For example, Fab fragments, nanobodies, and especially bispecific fragments binding to two antigens, which are, for example, expressed on the tumor cells as well as on a T cell. So you can actually bring the immune system very close to tumor cells in this way. So, from a pharmacological point of view, this field of antibody therapeutics holds promises of high specificity and affinity compared to the old strategies with small molecules, which are quite unspecific and causes severe side effects. It's also possible to have a deep tissue penetration with these antibodies, and they are quite, from a kinetic point of view, they are quite high. They have a high stability, and there are less toxic compared to, for example, old chemotherapies of cancer. So there are a lot of beneficial, you can say, properties of these antibodies or antibody therapeutics. So now I will go to this specific platform or technology which has been developed by Oblique Therapeutics. So as you see here in the title, is a novel lipid-based protein immobilization in microfluidic technology by Oblique Therapeutics. And the purpose is that this technology identifies promising epitopes or peptide sequences on membrane-bound and membrane-associated proteins by using limited proteolysis. And I will come back to that in the next slide, what it means. So the strategy here is to gain detailed knowledge about the surface which is exposed, and this surface exposed functional active epitopes or peptide sequences within a native protein. And a key thing here is that the proteins that are native, so it's possible to produce antibodies that are likely to affect the function of the protein. So this will facilitate the development of efficient antibodies and decrease the need for elaborate screening procedures by lowering the amount of antibody candidates. So here is the principle of this AbiProt platform. So the key thing here is that they are using proteases to probe, you can say, to probe the antibody binding site on a protein based on size similarity. So a protease has the similar size as the Fab fragment of an antibody. So a possible method to evaluate the surface topology of a protein is to restrict the activity of a protease to digest only the most flexible and exposed parts of a protein to which an antibody can bind to. So this is achieved in a very, very sophisticated control of the activity of this protease. That is, for example, to use low temperature, low concentrations, and short digestion times. So technically, as you see here in B here, it's possible then to immobilize the protein within a microfluidic channel, which will permit multiple rounds of this proteolysis under very controlled kinetics. And here you see here a combination of different kinetically controlled proteolysis strategies is possible then to release peptides for analysis by mass spectrometry, for example, to identify native surfaces, native epitopes or peptide sequences. Then from these peptides is then possible to produce antibodies, molecular antibodies, which can then be both agonistic and antagonistic antibodies. So now we return to the specific project here. So this is just a general description of this platform. So we are focusing much on pain, and this is why I want to bring up this project, because to describe the influence and the use of Fluicell's techniques in this, which has been very, very valuable in this project. So you may know that 20% of the world population suffers from chronic pain, and this is expected to increase with increasing aging of population. So from an economical point of view, then chronic pain has an enormous impact on economic productivity. So the price tag in the United States is $6 billion, which is more actually than cancer and heart disease combined. And indeed, there's a great unmet medical need. So only 25% of the patients receive sufficient pain relief from current treatment options. And the current pain medication, for example, the opioids, such as, for example, morphine, it has its limitation because it can induce drug dependence. It is very risky to use these kind of medications. There are quite few targeting which are ion channels here. Many of the treatments options are, for example, non-steroid anti-inflammatory compounds. So here we will focus on this ion channel or protein called TRPV1 or transient receptor potential valenoid receptor 1, which is an ion channel expressed in, you can say, primary afferent located in DRG neurons, and they are afferent, they are exposed in the dorsal horn of the spinal cord. So this channel is very, very important in mediating pain. And as you see here to the left here, it's a multimodality receptor. It can be activated by several, in general, activators such as capsaicin, endogenous ligands like NADA, protons, heat, and so on. So it's a multimodal receptor. So this has been a problem. I mean, previous efforts has been to develop small molecules for inhibiting this receptor in a clinical perspective to inhibit pain. But these molecules, they are bound to the whole receptors and blocked all of these modalities. Capsaicin-induced currents and proton-induced currents and so on. So this has caused, in phase 1 studies of patients here, it has been noticed that blockage of the heat sensitivity of this channel has resulted in hypothermia. And it has been reactions then in the phase 1 studies. So the purpose here with the project here is to just induce a kind of selective blockage of the analgesia part here, which can be induced by capsaicin, for example. So the purpose has been to develop a modally selective antibody. That is a selective antibody that retains just heat sensitivity in the channel but block the other modalities. So here's the title of the project that we have performed. So Oblique Therapeutics in Gothenburg, they have produced molecular antibodies against the TRPV1 channel, and we have investigated them by use of our models. So the purpose has been to test the pharmacological activity and analgesic effect of these anti-TRPV1 antibodies by using mainly two types of in vitro techniques, like the in vitro calcium imaging and the electrophysiological patch clamp recording. So if we start with the first one, calcium imaging, and now you have heard a lot about this very, very nice and fancy biopen, I don't think I have to go through all the details concerning using this biopen. But basically in this project, or in this method, we have used CHO cell, Chinese hamster ovary cells, and they have been able to overexpress human TRPV1 because it's very important to address here or investigate antibodies against the human TRPV1 here. And here we have used a calcium imaging technique, which is based on that we have loaded the cell with Fluicell, which is a sensor for calcium, for example, both calcium influx from outside the cell and also from, you can say, internal stores like endoplasmatic reticulum into the cell. It has been used a lot in, for example, muscle research and so on. And then we use that in combination with confocal microscopy. So here, as you see in the setup here, we can combine this, you can say, limited perfusion volume, where we just can, you can say, administer our drugs with a heat probe laser. So here, by using this heat probe laser, we can activate just a few cells in this culture here, and then see how the antibodies are affecting this, you can say, activation by heat. We have a complement here also. We have to, I mean, we were supposed to screen for like 14, 20 antibodies. So we need also a high throughput screening, a plate reader. And we have also used the calcium imaging technique here. So here we have been able to measuring the effects of several concentrations of the different antibodies in this plate reader. And here's the second method. It's related to patch clamp. And here we have used the same cells as in the calcium imaging experiments, the show cells expressing the human TRPV1. And here, as you see here, the patch clamp is about to, you can say, attach a cell to a pipette tip. And this, you create, you can say, a giga seal between the cell and the pipette tip. And then you have further suction here, and you will disrupt the membrane, so you have access to the internal part of the cell by the electrode solution here, normally consisting of a high concentration of potassium. And then you can position these cells in front of the tubes here, the Dynaflow tube. Maybe I've talked about this before. And it's a very effective model here. And the beneficial things here with this that it was mentioned in the previous talk is that you can perform quite fast experiments. I mean, this patching here of this cell here is very sensitive. And the patching holds for example, for 20 minutes or something like that. You see also, you can also prepare a lot of solutions here, and it means that you can do very, very effective and fast concentration response curves by using this Dynaflow technique. And also another thing there, perhaps you have mentioned that, is that the flow from the tubes here is laminar, so you have very low mixing of the different, you can say, buffers or drugs here, which is very beneficial. So you can get very nice responses from the different drugs that you're applying to this patch clamp cell. So we have added capsaicin, NADA, and together with antibodies. And here you see one example of the trace here. With NADA alone, then you can activate an inward current here, which is mediated by calcium. And then you add this antibody here, OTV4, together with NADA, and you see that OTV4 is blocking the response by NADA. And then you can, by flushing away OTV4 with this very effective system, you can retain the response from NADA alone here in the next response here. So here are a kind of summary of the results here. So first we tested like 14 polyclonal antibodies on these human TRPV1 induced responses. And we used two actually small molecules as controls here. Is the Mavatrep binding to, in general, to TRPV1, and this AMG 517. And you can see that they are quite effective in both blocking the capsaicin-induced currents as well as the heat-induced responses in these cells, which are TRPV1 mediated. But, which is very interesting here, is that the antibodies produced a selective inhibition of just the capsaicin-induced activation of a TRPV1 channel, but what was ineffective to actually block the heat activation of these channels. And this is on human TRPV1. So we did the same experiments in mice, on mice DRG neurons, these so-called pain neurons, and then we were unable to see any effect of these antibodies on mouse TRPV1, so to say. So here are more results. Then we selected a couple of antibodies from the previous studies here with the polyclonal antibodies, and they produced monoclonal antibodies. So here we can also see the modality that they are also modality selected. So first here we see binding studies here, that one of them, OTAB1, is have a very great affinity in binding to the show cells expressing these TRPV1 channels. But if we do not express the channel in the CHO cells, we cannot see this effective binding. And here we also see that O2 AB1 is very, very effective in blocking capsaicin-induced currents here. And while Mavatrep, of course, and Amgen 517 also are effective in blocking these currents. But we didn't get any inhibition of the heat. So as I said before, also these monoclonal antibodies that were very effective, they were not effective in blocking the heat, while these control molecules also were effective. And then we went again to made concentration response curves here. So we went on with this OTAB1 and could see that we had a concentration response relationship here. If we increase the concentration here, we saw a better effect on the capsaicin inhibition. And this was repeated by NMI in Austria. They also did the same experiments here and could see that capsaicin could be blocked, capsaicin-induced currents could be blocked by this OTAB1. So in summary, we can here then demonstrate so to say modality selective monoclonal antibodies. For example, this OTAB1, which is targeting the human TRPV1. This is the first pharmacologically active monoclonal antibody targeting this ion channel, as well as the first rationally designed antibodies against an ion channel. Thus, by using this novel platform for antibody production, then Oblique has developed monoclonal antibodies targeting this human TRPV1, which is a very important target that has failed, as I said, using small molecules approaches. So due to this complexity of its activation mechanism, most compounds targeting TRPV1 have produced adverse side effects, such as hyperthermia or loss of heat sensation. So a modality-selective monoclonal antibody inhibiting capsaicin activation, but not heat activation, could then, as we think, offer a safe solution to improve the treatment of pain. So thanks to all these technologies that we have been using, we have been able actually in detail to study the effects of these antibodies on a very, very important target here, the TRPV1 channel. Thank you very much.
Thank you, Professor Jardemark. So is there any question first from any of the speakers to start? And maybe we can start with any of the participants.
Shall I take away this the slides?
Yes, you can. Or you can. Yes, absolutely.
Yeah, so I can ask one thing. So,
yes.
Yeah. So with their TRPV1, assuming that the mouse and the human one are fairly similar.
Yes.
So if I'm not mistaken, the capsaicin receptor is an internalized receptor, and the proton one is there's different locations for essentially where the binding parts are. Is that correct, right? So
yeah, right.
So
but they are, yeah, I know what you're thinking about. Yes, yes. Mm-hmm.
Yeah. So I mean, are there like different, let's say with the binding of the antibody fragments, are they only externally, binding, or do you have to also look at the transport mechanics of the antibodies to the binding sites to, activate or deactivate their receptors?
Yeah, that's very interesting. I mean, maybe you know about the previous experiments in Uvorva's lab that they have Karina and Trukulia and Uwe, they have used internal antibodies for binding the internal binding sites, so to say. But here we believe also that there are actually external binding sites for capsaicin as well, because we have a very quite fast response here. And also, which is very important, is that the antibodies, they cannot, they cannot enter the cell in this setup.
Okay, cool.
Yeah.
Thanks.
But of course, you can have, it's very complex. So you can have conformational changes by adding capsaicin in different ways. I mean, allosteric modulation and so on. We try to investigate that, the mechanisms here, whereby antibodies are blocking.